Hello, everybody. Welcome to this week's Infection Control Matters. My guest this week is Dr Ginny Moore, who's the scientific lead for the built environment at the NICUs. And we're going to be talking about a topic we've actually had on before when Alicia Demirjan talked about staph capitis. So, Ginny, you've just had a new paper out. Thanks very much for agreeing to talk to me about it.
No worries.
Okay, tell me the background of the paper, why you went out looking in the environment, and then we'll discuss techniques if people are interested in doing that sort of stuff and where we might go with it.
Okay, so I think probably Alicia probably gave you a good overview of staph capitis when she was talking to you. But just as a recap, during summer 2020, UKHSA was alerted to a perceived increase in invasive infections caused by methicillin-resistant Staphylococcus capitis in hospitalised infants. A national incident requiring a UKHSA standard response was declared in June 2021, and then a number of task and finish groups were established to focus on different aspects of the response. And I was involved in the task and finish group, which focused on the healthcare environment, and specifically the potential for staph capitis to colonise and persist within neonatal units. So basically understanding if and how this organism can survive with this environment would then help inform perhaps preventative or infection control measures.
Okay. So as part of this then you went out sampling in the southwest of England. Can you tell me how that went and how you decided where to sample and a bit about the zones you picked as well, because that was quite interesting.
We took environmental samples from 4 different neonatal units, 2 of which at the time of sampling had known colonisations on the unit and 2 without. And so we used a zonal analysis approach, which basically focused on the baby as the potential source of infection, with the hypothesis being that the surfaces immediately surrounding the baby would be more likely to be contaminated. And then we took samples in zones increasing diameter away from the baby. So we focused initially on the neonatal unit, the neonatal incubator, then the immediate bed space, so you can imagine moving out, and then further within the NICU bay itself. So perhaps more commonly used items of equipment like the milk warmers. And then we also then moved out of the bay in which, say, the colonised infant was, to take samples in staph-only areas or areas where the parents may use.
Okay, and so these were multiple incubators in one room, or was it one incubator per room in those settings?
So in the settings that we visited, there were multiple incubators in one room.
Okay, and what sort of spacing between them, just as a matter of interest, really?
It did depend very much on the unit. So that was one thing that was obvious. I think the units differed, particularly in the ones that we visited, because we visited some very modern units, in which case there were quite, a couple of meters space between the incubators, to some much older units where obviously the just the environment itself was a little bit more cramped. So it did vary depending on unit.
Okay. Now, you said you had some where you had known cases and some where there weren't any at that time. Do you know how long it had been since a previous case, or had they had none at all, no history at all?
So one of the ones we visited, for everybody's, in terms of the knowledge available by everybody there, they'd had no cases. And in another one, there'd been 12 months between cases. Okay.
And were they actively looking for it? Were they screening, or it just would have come up as a clinical sample, something like a blood culture that they would?
Yeah, so my understanding is that there was no screening. My understanding is it would have just come up as when a specimen was taken.
Okay.
A blood culture or something, yeah.
Yeah, and it's generally only blood cultures that are speciated, really, for the coagnac staph, isn't it? So that's why it can slip under the radar.
Yeah, I believe so.
Yeah, okay. Can you talk about how you actually did the sampling? Because people are interested in that sort of methodology.
Yeah, so as you know, there's a variety of ways you can choose to sample a surface, and that can depend on the environment you're sampling. But in this instance, we used just nylon flock swabs, and resuspended the swab in a liquid amine solution and then plated just onto blood agar.
Okay.
So it's quite a resource intensive procedure, because obviously once blood agar detects everything. And so we did do a lot of subculturing and moldy toffing to identify staph capitis. Alongside this particular study, we did spend a bit of time looking to see if we could utilize any of the chromogenic media that are currently out there to make our lives a bit easier. But for the purposes of our study, we cultured everything that we could and then identified everything that we could.
And actually, did the chromogenic agar have potential, do you think? I was thinking about if you had would this be something a normal clinical micro lab, even though they're not accredited generally to do environmental samples, would they be able to use these techniques to check their own environment?
I think so, and I think perhaps what the chromogenic agar might allow us to do is actually make screening out some organisms or some colonies easier. So even though it might not identify the target organism, staph capitis in this case, it might help us not bother with a whole heap of colonies. So it might make our life easier that way.
Because I can remember using chromogenic agar for MRSA to pick out MRSAs. It was the same sort of principle, is it?
Yeah, very similar.
Okay.
Yeah,
what
very
did you
similar.
find then? Tell me what you found.
So we sampled, I believe, she says, 173 surfaces throughout the 4 NICUs. And in fact, that was actually 5 visits, because we did actually go back and sample one of the units twice. And 40 of those, so about 21%, harboured staph capitis, and 30 of those, so 75% of those staph capitis isolates identified were actually the methicillin-resistant NRCS-A clone, which is the clone of interest in this case.
Mm-hmm.
And we did recover staph capitis from surfaces across the NICU setting, but the NRCS-A clone was rarely recovered from outside what we're calling zone A, so the one immediately surrounding the baby.
So zone A is just the inside of the incubator, or inside and outside and just the whole of that piece of kit?
Yeah, so inside and outside the incubator.
Okay.
Yeah. So what I should say perhaps as well is that during this study, so we had the opportunity to sample incubators that had been taken away from the actual unit because they were due to be cleaned. And so what we were able to do was actually break down those incubators and sample areas within that incubator that if there was a baby occupying the incubator, we wouldn't normally be able to sample. So things like the mattress, for instance, or surfaces underneath the mattress.
Right, okay. But most of the sampling was done with the baby in situ, was it?
Yeah, most of it. Yeah.
Okay.
So just on those occasions, we were able to go inside the incubator as well.
Now, you found it in units that have obviously a patient currently there with, but you also found it in the units that didn't have. Do you know if actually there was much cross-transferring between these units? Was one of them as like a specialist centre and patients maybe were going in and out of it and back to these other units, or was there limited opportunity for that sort of transfer to go on?
No, so my understanding is that there was opportunities for babies to transfer between hospitals. Whether that was the reason for finding this particular organism within those units, I'm not sure. I mean, certainly finding or recovering staph capitis NRCS-A from uninfected babies has been documented previously. Groups in France have done a lot of work in this area and have found very similar results to ours. So it could just be that babies are colonized and just not causing any problems. So there's no reason to detect it. But certainly, to answer your question, there is scope.
Yeah, it's interesting though, isn't it? Because there's clearly something going on somewhere in that they found it, I think, first in the Netherlands or Denmark, and then all over France, and then all over here. And the number of countries, the list with the same clone in all around the world, there's obviously not going to be that amount of transfer of babies between the two. So I'm just wondering how's it getting into these? Because you're not finding it anywhere other than the immediate incubator and around the baby who's colonized. If I'm right, you're not finding it in the patient area. So, in the the family areas, so it's unlikely that the families are bringing it in, and why would they have the same clone anyway? Have you got any ideas?
Yeah, it's very interesting.
I mull it over a lot, and I no clue.
No, I mean there's previous studies that do suggest that it's not because the caregivers are colonized. I think current thinking might be that actually the antibiotics that are used within the neonatal units are kind of like providing some kind of selection pressure in terms of providing an environment which will allow these resistant organisms to perhaps proliferate or survive well.
I think it's got to be there to be able to do that, though, hasn't it? So how's it got there? I'm wondering if there's some piece of kit that goes into neonatal units that's very commonly used in neonatal units around the world, a bit like the heater cooler units that were coming pre-installed with mycobacteria. Is there something that's generally considered to be low risk, therefore no one's going to really worry about it, but it comes in that way?
Yeah, that's something not quite sure on yet. And I know there's a lot of work that's going on with by UKHSA colleagues and colleagues in Imperial, looking at perhaps the genetic diversity as well in terms of these organisms that are being detected, to get some indication as to how related they are.
Okay.
As well. So that kind of information as well will help.
Yeah. So you said you took apart some incubators without a baby in them, enabled them to get into areas that you wouldn't normally have got at with a baby there. What did you find with those? Did you find that you were getting plenty of positives from that area? And actually, how difficult was it to take these things apart to be able to get at those sampling areas?
So I think one of the reasons, one of the reasons why it could be that we're finding it on the incubator is that the incubator actually, it would appear to me anyway, has been quite a difficult piece of kit to clean. Yes. Once we did start to break the incubator down, you could see that there's quite a bit of nooks and crannies that bugs could quite happily survive in. So how easy was it to break down? I mean, certainly not trained, so if other people were doing that for us. But it does go to show that, you can only clean as well as you can dismantle an item of equipment.
I remember Peter Hoffman years ago, some colleagues marked one of those with an ultraviolet marker and challenged him to clean it, and he missed 6 out of 10.
Okay, yeah.
And if Peter Hoffman can't clean it, it's borderline uncleanable, I suspect.
Uncleanable.
Yeah. So, that's the issue, isn't it? I mean, you say that you weren't trained to do it. I'm sorry about the poodle in the background. I'll leave it out. You weren't trained to take it apart. Do you actually know if the staff who are cleaning those were really trained to clean that incubator properly? Because I've seen issues in the past where people haven't been that well trained to clean, and the product they're using is on and off in 10 seconds. It doesn't get any contact time. And in one outbreak of Acinetobacter I looked at, the only reason they got away with it is they were using hydrogen peroxide vapor on all of the incubators before going back into use. And when the chap went off sick for a period, they didn't get that bit, didn't happen, and suddenly they had a cluster of Acinetobacter cases. I mean, that's an issue, I think.
Yeah, no, I think you're right. And I think that, the cleaning protocols differed from unit to unit. And who cleaned the incubators differed from unit and unit as well.
Not uncommon.
So I think one of the things probably that this incident highlighted perhaps is that the need for a sort of like perhaps more national guidance in terms of cleaning within the neonatal unit.
You didn't happen to notice...
I think the thing is...
Sorry, I was going to say, you didn't happen to notice if they were all exactly the same incubator, or if you got lots of different brands, which would require cleaning in different ways as well.
Yeah, so in the main, I have to say that in the units, and obviously, we only visited 4, but in the main, they were very similar units, similar incubators, so they were quite similar. But I guess the thing is with environmental sampling is if you look, you can find, and that's not to say that because you find that becomes a risk. Yeah. So recovering or isolating an organism doesn't necessarily mean that that's going to cause any issue. It could simply be, it's an indication that perhaps cleaning could be improved, or it's an indication that a surface is difficult to clean, or it's an indication that people like me swing by with a swab, take a sample just before somebody has cleaned.
Yeah. Yes,
and
that's
had I
right.
sampled 5 minutes late, sampled 5 minutes later, it would be clean. So,
I was just wondering that really, because if the babies are colonised, then you're likely to get contamination. But the fact that you'd sample some that weren't in use and that were ready to go, that had been decontaminated, ready for the next one, that indicates that maybe the cleaning isn't effective enough. I mean, is there some way of auditing the cleaning that you might be able to make a recommendation on? Because doing micro is very time intensive. Results take ages to come back and that sort of stuff. I mean, were any of these units using any form of audit process to get some sort of assurance that the incubator had been cleaned well?
No, not in the units that we'd sampled. I think we did recover this organism after one of them had been cleaned, and that could well have been due to some kind of cross-contamination during cleaning.
Mm-hmm.
Another one, I think, when we're going right down into the depths of the incubator, that just could be because it's very difficult to clean. I think one of the areas that we want to look at in a bit more detail, in all likelihood to rule it out as any kind of risk, but the fan within the base of the base of the unit.
I mean, because if you look at desktop fans around the place, they're normally covered in what we call gunk. Were they visibly clean, the fans, or because that certainly would disseminate again, wouldn't it?
Yeah. So again, it differed between unit. Now that could be because different in between age of incubator, or it could have been due to differences in cleaning protocol.
Okay.
But certainly, I think that's one area that would be quite interesting to investigate a bit further.
Yeah.
Like I said, just to rule out anything more than anything else.
Yeah. Okay. I mean, how would you do that? Would you test the air coming out of the fan to see if there's dissemination, or would you actually sample the fan blades itself?
Yeah, I think here at Porton Down we have the facilities which we can use. So I think we could go full worst case scenario and artificially contaminate a machine, and then see if we can detect the marker in the air with some specialist air sampling equipment. So that would be the aim.
I'm not familiar with the incubator, but presumably if it really was coming out air-wise, does that blow into the incubator? Does it blow away from the incubator? In which case you would expect to see it a little bit more widely disseminated, which you didn't really find.
Yeah.
Or is it actually bringing cool air in towards the baby? I'm not sure.
Yeah, so I think my understanding is it's a way of distributing the humidified air.
Okay, so it's towards the baby rather than away.
Yeah, and I think that's another interesting. I actually think that the neonatal incubator is really quite an interesting piece of kit. The fact that the humidified air, the higher temperature that can be within the unit when the babies are very small. How does that affect the survival of any organism, really?
Yeah.
not just Staph capitis.
I was also wondering about the condition of the incubators, the plastics and that sort of stuff. Were they getting damaged or crazed from use of different chemicals that, because people have different protocols, that sort of stuff. And then when you get damaged, they become much more difficult to clean. But were there any obvious issues like that with them that you spotted?
So certainly it was interesting talking to some of the staff as to the difficulties that they found with some cleaning products. So some did bring up the fact that some cleaning chemicals can damage particularly the plastic, the plastic covers, which makes it difficult for them to see through as well. It's not the practicalities of using the thing, not just cleaning it, but certainly we didn't notice that, but that wasn't the focus of the investigation. But certainly anecdotally, some of the cleaning chemicals were highlighted as there's a bit of material incompatibility going on. Yeah,
sure. So, it'd be interesting to do a bit of qualitative research then on what the barriers were to staff actually. Do they actually feel they've cleaned an incubator well? How long does it really take to clean an incubator, and how long do they get to clean the incubator? And I suspect they are two different numbers.
Absolutely.
Yeah.
Okay. No, I completely agree. Yeah. Practicalities against efficacy. Yeah.
Efficacy, yeah. So any other pieces of work going to spin out of this now? Or are you going somewhere else with this particular piece of work, or is this the task and finish group done with this area?
So I think this is probably, this was what a main focus of the task and finish group. The UKHSA have stood down the incident. Like I say, it would be nice to look at the cleaning of the incubator. So that's something that would be quite keen to look at. But also, as you just pointed out, get kind of like the opinions of and a real life experience of incubator cleaning.
Yeah.
And perhaps, look at, for example, it would be nice to engage with some of the manufacturers of incubators as well, just to...
Yeah, I was just about to ask that, have they shown any interest in this work? Because they're making the kit that is actually difficult to clean, and how much human factors do they put into their design processes when they're making something, I've looked at pulse oximeters recently that you put on your thumb, and you squeeze it with your fingers to actually put it on, and there's 3 nice little grooves on it so that it doesn't slip. Well, but what pressure is that going to be at? But what you're doing is making it uncleanable. Yeah. When it could be perfectly smooth, because you're only squeezing open something that's going on somebody's thumb. It's not like you need a vice or something like that to do it. So there's a lot of design pieces that actually make something much more difficult to clean. And so it'd be interesting to see if the manufacturers were to prick their ears up and say, we'd like to do a bit of work on this as well.
Yeah, no, absolutely, definitely. That would be nice, because I think it doesn't have to be huge. Well, I say that not knowing how much any of this costs, but it would seem that it doesn't have to be huge changes to facilitate cleaning.
Or if they're designing the next model,
making it easier to clean. Yeah,
okay.
Yeah, we're just making it easier for staff to clean. Yeah.
Okay, fascinating stuff. I was still no nearer answering my question though, where the hell's it coming from? But there we are. It will come out in the fullness of time, hopefully. And everybody go, damn it, I had an idea it might have been that. But there we are.
Yeah, any ideas?
Yeah. Answers on a postcard to the following address. Yeah. Okay. Okay. Well, thanks very much, Ginny. I really appreciate your time. Fascinating study. Nice practical stuff. Thank you very much.